Between 2600 and 1900 BCE, the Indus Valley Civilization spanned northwestern South Asia, characterized by elaborate drainage systems, standardized weights, and sprawling urban centers. Despite its immense scale, the genetics of its builders remained a blank spot. Hot, humid climates rapidly degrade ancient DNA. At the Rakhigarhi Cemetery, one of the largest known sites, researchers attempted to extract genetic material from 61 different skeletons. To ensure the results weren't modern contamination, the team had to screen the extractions for ancient DNA damage, specifically looking for cytosine to thymine molecular mismatches at the ends of the sequences. Ultimately, only one sample passed the test. Recovering a single viable genome from this population allows us to test long-held theories about human movement and the origins of agriculture in South Asia. The successful sample carries the designation I6113. For decades, a dominant historical model called the Anatolian hypothesis posited that both farming economies and Indo-European languages spread into South Asia through a massive migration of early farmers from the Middle East. Looking at this topographic map, the theory suggests these groups moved east across the Iranian plateau and down into the Indus Valley. If this sequence of events is accurate, we should see the genetic signature of Middle Eastern farmers present within the I6113 genome. Instead, the sequencing results show zero evidence of these expected markers. To identify who the people of the Indus Valley actually were, geneticists construct an admixture graph, a mathematical model that maps phylogenetic relationships to trace exactly where these ancestral lineages originate. This graph illustrates the ancestral makeup of I6113. It reveals a mixture of two main lineages, one related to Southeast Asian hunter-gatherers, and another linked to early populations from Iran. Basing conclusions about an entire cosmopolitan civilization on a single sample is statistically precarious. We need independent validation that this profile was widespread. We find that validation in this map. Researchers had previously sequenced remains from Gonur in Turkmenistan and Shar-e-Sokhta in eastern Iran. Within those excavations, 11 specific individuals were flagged by statistical models as stark anomalies compared to the majority populations in those ancient towns. When compared, the genetic profile of those 11 anomalies perfectly matches the profile of the single Rakhigarhi sequence. Coupled with material culture artifacts linking them to South Asia, the data points to these outliers being migrants from the Indus Valley Civilization who had relocated to foreign cities. Together, these 12 individuals form a consistent genetic gradient known as the IVC cline, providing a robust, validated baseline to test other historical chronologies. Looking at this temporal chart, we see that during the mature phase of the Indus Valley Civilization, there is zero evidence of steppe pastoralist ancestry in the IVC cline. This contrasts sharply with the demographics of modern South Asians, who carry a prominent genetic component inherited from those steppe pastoralists. Since the trait is ubiquitous today, but absent in the Rakhigarhi and outlier samples, the admixture had to occur after these individuals lived. This provides a definitive temporal boundary. Massive movements of steppe populations into northwestern South Asia did not happen before 1900 BCE. With their arrival securely dated to the post-IVC period, steppe populations are mathematically ruled out as the instigators of early agriculture in the region. This admixture graph exposes a second critical absence in the data. The IVC cline carries negligible Anatolian farmer-related ancestry. By 6000 BCE, all sampled ancient genomes from crop farmers on the Iranian plateau exhibit significant Anatolian farmer-related admixture. Because the IVC lineage completely lacks this Anatolian signature, they cannot be the descendants of the early farming groups moving eastward from the Zagros Mountains. To locate the actual source of their Iranian-related ancestry, we must trace the phylogenetic tree backwards into the Pleistocene. The model confirms that the Iranian-related lineage feeding into the Indus Valley split from the main Iranian plateau lineages before 10,000 BCE. At 10,000 BCE, human populations were strictly foragers. Crop farming would not be invented for several thousand more years. The genetic link between early Iranians and South Asians stems from a deeply ancient divergence of hunter-gatherers, rendering the theory of a sweeping Middle Eastern farmer migration biologically impossible. The Rakhigarhi genome presents two hard constraints. It contains no steppe pastoralist DNA and it contains no Anatolian farmer DNA. The ancestral populations residing in South Asia following that 10,000 BCE split remained genetically isolated from the later agricultural innovations occurring in the Fertile Crescent. Therefore, the transition from foraging to the complex, granary-supported urban centers of the Indus Valley Civilization was achieved by local populations developing farming practices independently. This stands in stark contrast to the agricultural history of Europe, where genetic sequencing confirms that farming was introduced via large-scale physical migrations of Anatolian populations. The data from individual I-6113 confirms that northwestern South Asia operated as its own fully independent cradle of agricultural and urban development. This biological data also resolves the linguistic branch of the Anatolian hypothesis, the argument that Indo-European languages were carried into South Asia by those same migrating Middle Eastern farmers. With the genetic divergence definitively dated to 10,000 BCE, there is no physical mechanism to link the onset of early farming in the Indus Valley to the introduction of Indo-European dialects. Instead, the genetic record aligns with linguistic evidence pointing to a later transmission via Central Asia in the first half of the second millennium BCE. This timeline maps precisely onto the arrival of steppe pastoralist ancestry, which we established strictly postdates the mature Indus Valley Civilization of 1900 BCE. By successfully sequencing a handful of degraded DNA molecules from a single petrous bone, researchers dismantled two intersecting models of ancient human history. The Rakhigarhi genome demonstrates that the total absence of expected genetic markers can be as definitive as any artifact in proving the exact sequences of human migration.
Distorted narratives can warp a civilization's future. Alignments in the sky never lie. Subscribe to step into the next forensic file. See the record corrected.